So our next speaker is going to be Dr. Agata Wolna. She is currently a postdoc here in F. Fedorenko's lab, and she is interested in the neurological basis of language. Before coming here, she studied at the Jagiellonian University in Krakow, where she was also interested in language, but more in terms of bilingual speech production and also cognitive control. So we'll go ahead and get started.
Well, thank you for this kind introduction, and I'm really happy to be here, and I'm honored to have been invited to speak at this wonderful event. And I would like to take this time to give you a short overview of the work of the Fedorenko Lab here at BCS at MIT. And I would like to focus specifically on the functional precision mapping, which is a method that we use to study language and other cognitive systems in the brain. So I really hope that this talk will kind of follow the path set by my preceding speakers in terms of showing some tools and some solutions that we can use to build cumulative and reproducible science across different sites and across different populations. So to start broadly, when we think about cognitive neuroscience, this is like a domain that asks questions about relationship between various cognitive mechanisms and the brain. So I guess we can say that its ultimate goal is to create a neurocognitive model that allows us to understand how cognitive mechanisms and cognitive systems are represented in the brain and how they interact with one another. And, well, this is a very ambitious endeavor, and one of the challenges, well, one of the many very difficult challenges that it has to face is related to the fact that human brains are inherently different from one another. So one big source of this variability comes from the anatomical differences between different individuals. And they relate to the size, the precise shape, and the proportions of different structures of the brain. Like Satrajit commented, like even in monozygotic twins, you see differences in how the brains look like. And, well, on the one hand, today we have tools that allow us to precisely reconstruct individual brains, so account for these differences in individual anatomy between participants. But when we want to do a functional study, so when we ask about the correspondence of function to anatomy, what we have to do is we have to register these individual brains to some kind of standard template, because we need some kind of common space to be able to make inferences about, well, about cognitive system, how cognitive system looks like at the population level. And by doing so, by registering individual brains to these common spaces, and there's a lot of different common spaces, no matter how advanced our registration algorithms are, there's always going to be some smoothing of signal. We're always going to lose some information, and we're always going to have to average and get rid of some kind of information that allows us to model this idiosyncrasy in individual brains. But I don't think this is the biggest problem we have, because another huge source of variability in modeling neurocognitive organization of the brain is the fact that people also differ in the functional organization of the brain. So this is an example of three different participants' language systems. So you can see that they map roughly on the same macroanatomical areas. So in all of them, we see this frontotemporal activations, but, well, the details are vastly different. So the peaks of activations differ, the size of these systems differ, and the precise anatomic, topographical localization and the boundaries of the system are also different between individuals. And if we register that to a common space, what happens is, unfortunately, that we're going to not only lose the information that's related to differences in anatomy, but we're also going to blur over the boundaries of the functional systems. And we're no longer sure if this particular voxel in the brain of person A corresponds to the language system and not another system compared to the person B, for example. So today I would like to show you a solution that we use and convince you that precision mapping that's based on functional localization in the brain is, well, a possible solution to this problem. So, if you want to use this precision mapping approach that defines the systems in the brain functionally instead of anatomically and on individual subject level, instead of relying on some kind of group level and population level representations, It's really pretty simple. What you need is something that's called a function localizer task, which is an experimental paradigm which allows you to robustly and in a very broad sense define the cognitive system of interest. So in our lab, to identify the language network in the brain, we use a contrast between reading sentences, so these are well-formed full sentences, versus reading lists of meaningless non-words. And this paradigm has been validated in different modalities and in different languages and, well, in many different task versions. And we now know that it allows us to very, very precisely localize the language network in each individual participant that we scan, separately. So instead of, we don't have to rely on their anatomy to define the regions of interest that we want to study, we can actually define this region of interest functionally for each participant separately. And then we can use these language systems that we identified as kind of regions of interest analysis and ask questions about what do they respond to. So we can test the responses of the language system to different language conditions, but we can also test the responses of this system to different non-necessarily language tasks and ask what is the functional profile of the system we are studying. Okay, so to sum up, this approach allows us to model individual variability by at the individual subject level, so it kind of overcomes the challenge of inter-individual variability and topographical organization of functional systems in the brain. But it also allows us to keep the analysis space constant. So instead of having a constant anatomical space, we have a constant functional space that we can keep between participants and different populations and between different experiments. Because when we define the analysis space functionally using the same robust localizer tasks, we can say with very high degree of certainty that we're looking at units that are meaningfully doing the same in the brain, even if they're not exactly, if they do not show exactly the same topography. So I would like to tell you now about the study that we recently completed, and that's hopefully going to be published very soon, and that showcases some advantages of functional localization. So in this experiment, we reanalyzed a very large data set of participants who all completed, so 772 participants who over several years, actually I don't know, almost 20, completed the reading language localizer task. And we were interested in seeing what regions in the brain we can find that robustly and that show some responsiveness to language, and what are the characteristics of these regions. So, so what we did is, now it's going to be a little bit confusing, but we overlaid all of their statistical maps for the language localizer contrast in a common space, but not to make any inferences, but to create a probabilistic map that we then parcellated to get this really nice map of parcels here, and you can think about them as search spaces. So this is a search space in which you have a high probability of actually finding the individually defined language systems in the brain. And this is useful because sometimes, we don't want to use a region of interest that's defined at whole brain. We want to kind of be able to go down a little bit and ask stuff I don't know, are there any differences between frontal component of a system that we're studying, the temporal component, the cerebral or subcortical components, and so on and so forth. So this is exactly what we did. In the system, in each of these search spaces, we identify the language system individually for participants, and we checked if it responds to two different language conditions. These are the red bars here. So this is a reading and auditory language tasks. And we also checked if they are selective to language over a difficult executive functions task. So if we see some mixed selectivity there, or are these really kind of regions that are biased to process language? And as an additional thing we did that I also wanted to show you, we also compared how this approach how compares to kind of traditionally used ROI analysis, in which we just take some region in the brain that's defined in some kind of standard space, Harvard Cortical Atlas, or the standard free surfer, which is the DKT Atlas, or even very fine-grained Glasser parcellation, which is function/anatomically derived thing. And and we just basically compared how does it compare to our functionally defined regions of interest. Okay, so should we do it here? Yeah, okay. So the first finding is that we kind of replicated a large body of findings that show that language robustly activates the left frontotemporal system. And we also see very nice, well, a little bit smaller, but still a very nice response to language in the right homotopic areas to that system. But apart from these kind of like canonical language areas, we're also able, like, because of the sample size and because of like this probabilistic map threshold at many different levels to try and find these, to identify regions, for example, in the frontal medial cortex or in the ventral temporal cortex, and also in the cerebellum, that also show this this selectivity to language over other non-linguistic tasks. So it doesn't mean that they're selective to language. It means that there is some degree of selectivity over different tasks. That it would require to run a much more kind of complex battery of things to say exactly what they're doing. But we see that they definitely do something for language. And interestingly, we also identified a bunch of regions in the occipital cortex that showed a response to the reading language localizer contrast. It's actually one of my favorite results here. So you can see the first 2 bars in each plot are the reading localizer contrast. And if you only run this and if you average that between participants, you might kind of think, okay, so there's like language in the occipital cortex, like that's really cool. But these responses do not generalize to a different language task. So it's not high-level model language processing. And what we also see is that this region respond much more strongly to a non-linguistic executive function task. So they're not really language regions. They, for some reason, respond to one of the paradigms that we're running. OK, so now let's move on to the advantages of the functional localization. So how does the functional localization make this endeavor easier? So here I would like to show you like a comparison of 2 different regions of interest analysis that we run. And we took 2 different regions of interest from the Glasser oscillation. So it's going to be one region of interest in the left IFG and one in the posterior temporal cortex, which are both like very kind of classic-y language regions. And on the left, we're going to see functional profiles of these regions if we just take the entire ROI and average over all the voxels. And here we have the same ROIs, but we just take the top 10% of most responsive voxels to our language localizer contrast. And well, here are the results. So this is the left IFG. And what we see is that it shows the response to language, but it also shows the response to a non-linguistic domain general task, which is kind of consistent with some of the literature that proposes that this region actually shows mixed functional profile. So it supports language, but it also supports other functions. And on the other hand, in the temporal cortex, we see very strong response to language and no response to the domain general task. But when we look at the individually defined regions of interest, well, the first striking thing is that, well, the effect sizes increase. Our statistical power kind of like goes through the roof when we do that. And then what we also see is that in the left IFG, this mixed profile is gone. So it seems that there are like 2 different kind of functional systems that fall inside this region of interest that lie side by side of one another, but without actually functionally localizing them. We're going to blur everything together, and we might kind of end up making kind of false inferences that there is some mixed functional profile of the left IFG. They're like just different systems that are very closely related in the space. And we also did that analysis for all the regions of interest in 3 different atlases. So here on the left side, you see all the regions of interest that showed selective response to language over this non-linguistic task. And the color coding corresponds to like how strong that selectivity to language was. And on the right side, you see the same, but for the functionally defined ROIs within these macroanatomical regions. And what we see is that with functional localization, we get much more regions that show that selectivity. So potentially, we're kind of expanding our search space. And the selectivity is also much stronger. So I hope that shows some advantages of functional localization. And now I'm going to try to convince you that using the tool, this tool is not only good because, well, it allows you to, well, draw correct inferences and precisely disentangle different systems and increase statistical power. It actually works really well, like with the language system, it works really well at, I don't know, like individual subjects. But if you scan 12 people, it basically the results just stay very, very constant. It seems that the functional organization in the brain is not like very variable between participants, just the topographical localization of this network, of these networks that varies. But another advantage of functional localization is that if we define the analysis space functionally using the localizer tasks, it kind of creates a window to robust cumulative science, because what we can do is across like the span of years and in different research groups and across different populations, we can define the same system and we can ask a diverse set of questions about this system. So here I'm going to show like a summary from like a recent review paper from our lab that basically summarizes some part of this huge endeavor for the language system. So yeah, so if we look at the responses of the language system over like really, I don't know, this is like, we're at over 1,000 sessions right now, like over 100 experiments. So in all of these, we see very robust response to reading language, to auditory language. Then we also see some smaller but non-zero response to speech perception conditions. So there seems to be some overlap between functions of these networks and speech perception. But we, for example, don't really see any overlap, any engagement of the language system in music perception or in numerical cognition or in executive functions. It does show some response to processing of semantic events, especially the temporal cortex seems to be engaged in these things. And there is also some engagement in high-level social cognition, which is a very interesting thing because unlike this claim that language is made to kind of, that the function of languages allow us to think, this kind of hints that the function of language is communication. And it's actually human social interaction primarily. And then another thing that we can do with functional localization is we can use it to disentangle between two different systems. So this is an example of a study on 88 participants that we did in the lab. And we made all participants kind of complete the language localizer contrast to identify the language system, but also another localizer that allowed us to identify the speech motor production system in their brains. And one thing that we can do is we can identify these both systems, whole brain, and then look at their overlap or some kind of, I don't know, spatial correlations between these systems. And I'm not going to show you these results, but they're strikingly different. I mean, there is very little overlap between speech articulation and high-level language. But then, I mean, one might think, well, there has to be convergence somewhere. So what we can do is we can look at the functional profiles of these two systems. And well, in the language system, we see very robust responses to language conditions. We see, well, a little bit above zero response to the articulation, to the speech articulation condition, which is meaningless syllables, but this is very low. We don't really see any non-zero responses to repeating non-words, to repeating meaningful words in isolation. And this is like a very fun task. This is a verbal fluency task. And the language system shows a very clear preference, like very clear response to the lexical access difficulty in this task. And in the contrary, in the articulation system, we see very robust responses to articulation. Like, note that the scale is completely different here because, I mean, like, it's a very, like, motor responses in the brain are very robust. And we don't really see the responses to language. So it may seem like this is a significant response to the auditory language processing, but this effect is mainly driven by the fact that in the superior temporal cortex, which is a part of the language system as well, there is some kind of overlap between, well, perception of speech and articulation. And what the system does respond to is to the difficulty of articulation. So hard non-words and foreign words are more difficult and more engaging than easy non-words and native words. And very interestingly, for the verbal fluency task, we see a completely opposite pattern. So it responds to the amount of output. It doesn't care if that was difficult, like the word was difficult to found or to retrieve. It just cares of like for how many words you actually produced. So I'm wrapping up, I promise. So I hope that I managed to convince you, or at least get you interested in functional localization paradigms, and show you that this is a tool to potentially address the challenge of brain heterogeneity. And it not only increases the statistical power of our analyses, but it also allows us to precisely disentangle and characterize different functional systems in the brain. And because we're working in the shared functional space, not anatomical, it doesn't really matter that much if we use the same scanners. It doesn't really matter if we use scanners at all. Like there are people who do that in ECOG and in EEG, and it also works as well. And we can actually make very nice inferences between these different methods using functional localization. Okay, so here I would like to thank all the members of the Fedorenko Lab that contributed to this huge data collection over the years, especially the team that worked on this paper that I was showing you with me, and some collaborators from Poland, here they are, that contributed to the data collection on the speech articulation project. Okay, sorry.